Hello everyone and welcome to another class of Physiology for Engineers with Professor Gonzalez-Fernandez at Lehigh University. Today we are talking about the chemical synapse. So previously we have seen the membrane potential, we have seen the graded potential and the action potential. So let's say that the action potential happens and we finally arrive to the axonal terminal and we have these vesicles that are loaded with all these neurotransmitters that need to be sent to the next cell in order for the nerve impulse to arrive to its destination. So we are going to talk about that today, and we have three different elements that are really important. The first one is here the presynaptic neuron, which is always going to be a neuron. Here we have the postsynaptic cell. So the postsynaptic cell can be a neuron, it can be a muscle cell, or for example, like a smooth muscle, it can be cardiac muscle, or it can be a skeletal muscle, doesn't matter. And then we also have a gland. So we have these three options for the postsynaptic cell. And finally, here between the presynaptic cell and the postsynaptic cell, we have the synaptic gap. Whereas the synaptic gap is going to be the distance that the neurotransmitters need to travel in order to activate the postsynaptic cell, okay? So let's start how all this process happens. So we have different stages. So the first stage here is going to be the action potential is going to arrive here, right? So when the action potential arrives, there is this depolarization of the cell membrane. The cell membrane is more positive. And this action potential here is going to activate these voltage-gated ion channels for calcium, and this is going to pump calcium from the extracellular space to the intracellular space according or following the concentration gradient. Okay, so this is the first thing, and this is very important. Then we have a second process. So these vesicles here that are charged with neurotransmitters, they need to fuse with the cell membrane in order to be liberated into the synaptic gap. So this process is controlled by three different proteins that we don't need to know the specific names of the three of them, but this is called the SNAR proteins. Okay. So there's SNAR proteins that are like these chains here that are kind of like binding the vesicle to the cell membrane, and they are going to bring the vesicle closer to the cell membrane in order for it to fuse and then liberate all the neurotransmitters that are inside the vesicles. But this whole process is determined and is controlled by other two proteins. So the first protein here that we have is complexin. Okay. So the complexin is going to inhibit the SNARs to bring the vesicle closer to the cell membrane and to allow this merging of the vesicle with the cell membrane. But a part of the complexin, we also have another protein here which is called synaptotacmin. And the synaptotacmin is going to bind to this calcium So after binding to the calcium, there is going to be a conformational change, and the synaptotacmin is going to cleave here the complexin, and then the complexin is going to allow for the SNR proteins to bring the vesicle closer to the membrane, and for the vesicle and the membrane to fuse. So once this process of fusion happens, then the neurotransmitters are going to be secreted to the synaptic gap. And this will be, so this process is number 2, and this will be number 3. when all the neurotransmitters are released into the synaptic gap. Okay, so now the neurotransmitters are going to arrive here to the postsynaptic cell, and they can interact with two different type of receptors in the postsynaptic cell membrane. So the first receptor they can bind to, there are going to be these ligand-mediated ion channels. So they are very similar to the ones that we saw during degraded potential, right? So these are ligand-mediated or ligand-gated channels, right? So these neurotransmitters here, they are going to arrive, they are going to bind, and then they are going to action this ion channel, so for sodium, potassium, or chloride, as we have seen. And then we have another type of receptor. Okay, so this receptor is going to be a G-coupled or protein G-coupled receptor. And this is a protein G here. So once a neurotransmitter binds here to the protein G-coupled receptor, the protein G is going to be divided into 3 subunits: alpha, beta, and gamma. And these 3 subunits, they are going to have different effects on the postsynaptic cell. 2 effects that we are going to look at today are, for example, here in the membrane, there are other proteins that are called effectors. So these G protein subunits can bind to the effectors, and then the effectors can have another activation cascade further on in the cell behavior or the cell function. And these G receptors can also bind to ionic channels, closing or opening these ion channels in order to have a better response or an inhibitory response that we are going to look now in a sec. Okay, so this will be number 4, which is how the neurotransmitter arrives or what the neurotransmitter does in the postsynaptic cell. Okay, so we are going to look at 2 different concepts now. So we are going to look at the synaptic strength. So the synaptic strength means how efficient this is, so how efficient the neurotransmitter is to go from the presynaptic neuron to the postsynaptic neuron, and that depends on the presynaptic neuron, so for example on the presence of calcium, and that also depends on the postsynaptic cell, for example in the presence of ion channels or the presence of different receptors, the release of neuromodulators or also enzymes, right? And the second thing that I wanted to address here is the cleaning of the synaptic gap. Because in order for an efficient message to come and then like another one to come again, we need to clear all the neurotransmitters here for the synaptic gap so we can have the next message very quickly and very efficiently, right? So this happens through different mechanisms. So the first mechanism is that the neurotransmitters get reabsorbed into the presynaptic neuron. So that will be the first mechanism. We have a second mechanism, that is the postsynaptic neuron is going to release some enzymes that are going to destroy these these neurotransmitters. So that's another process of clearing the synaptic gap. And then, so this will be number 2. So number 3, we have the presence of glial cells. So these glial cells, for example, the astrocytes. Let's see if we can see an astrocyte here. These astrocytes are also going to clear the neurotransmitters. So this is another process. And finally, the fourth one is through the circulation, so blood here, that the blood is also going to clear the neurotransmitter from the synaptic gap, so we can have another chemical activation. So before I finish with this small lecture, I also want to give you some therapeutic targets and some drug targets. We can have different compounds that are going to mediate in different processes here in the chemical synapse. So one of these compounds it's very important because it has a lot of therapeutic applications is botox. So that's called, that's the botulinum toxin, which is secreted by the Clostridium botulinum. So this is a bacteria that grows in different fruits and vegetables. And be careful because it can be in your house and it can give you really bad disease. So what this botox or toxin is going to do is going to break here the snares. So if there are no snares, the vesicle containing the neurotransmitters cannot fuse to the cell membrane. Therefore, the neurotransmitter cannot be released. And botox is used, for example, for cosmetic reasons in your face with a polarizing effect. And that happens because botox breaks these SNAR proteins, so it avoids for the neurotransmitter to be released. Therefore, it avoids for the muscle cell to get excited, right? So that's one of the targets that we can use for therapeutic reasons. Another target that we can also do is, for example, here, the clearing of the neurotransmitter from the synaptic gap. So one of the methods here that we have seen is enzymes, right? So the postsynaptic neuron can secrete some enzymes to clear this neurotransmitter from here. So for example, if this was, this neuron here was secreting acetylcholine, then the enzyme here that will be degrading the acetylcholine will be the acetylcholinesterase, right? So this enzyme here, there is a gas that is called the sarin gas that has been used in World War II and World War I. So the sarin gas, what it's going to do is going to interfere with the acetylcholinesterase, so the acetylcholine is not going to be clear for here. So what is going to happen is that the acetylcholine is going to remain in the synaptic gap, so there is going to be a constant stimulation of the postsynaptic cell. And this is really bad, especially for our breathing and our heart muscle, because if our muscle is completely activated, then our muscle will stop working. So this is basically how the sarin works, how the sarin gas works. It avoids the clearing of the acetylcholine here in the synaptic gap, so there is an overstimulation of the muscle. Okay? Well, I hope you enjoyed this lecture, and I'll see you in next lecture explaining other concepts of the neural system.